Today we will discuss about these VCAM rules in the pancreatic cancer cells towards this Warburg effect. So what is this Warburg effect? Warburg effect is commonly known as the cells use aerobic glycolysis process for the energy productions, that is ATP production. However, it is inefficient process compared to the regular glycolysis process, as in the Warburg effect only 2 ATPs are produced, whereas in the regular glycolysis process 36 ATPs are produced. But this Warburg effect in the pancreatic cancer cells is helpful in other ways. Previously we have discussed how this VCAM helps in the migration and invasion of the PDAC, and here we will discuss about its energy requirement, what are the reprogramming, or whether this VCAM helps in the Warburg effect that helps these pancreatic cancer cells in the survival and proliferations, as Warburg effect is known to upregulates the survival pathways, that is P13K pathway. So in order to analyze this, they use the seahorse XF analyzers to check extracellular acidification rate. And when they use this silencing VCAM-1 in the PANC1 cell lines, in the KPN2 cell lines, they see that the silencing of the VCAM-1, there is a reduction in the Warburg effect that can be seen here in the panel A and panel B, indicating that the extracellular acidification rate are lower with the reduction in the VCAM-1. And also what they have find out is that intracellular lactate production and the glucose levels are also lower whenever this VCAM is been silenced. And the quantifications are shown in the panel C for the lactate productions. We can see that in the PANC1 and the KPN2, the lactates produced are significantly lower than the control case, and same results we have observed for the intracellular glucose level in the PANC1 and the KPN2 cells. On the contrary, when they use or when they upregulate or overexpressed this VCAM-1 using pcDNA 3.1 VCAM-1 in the PANC1 cell lines and the KPN2 cell lines, they see that increased extracellular acidification rate. It can be seen from this seahorse XF analyzers for this PANC1 and KPN2 cell lines, and these things are quantified. These things are not quantified in this figure, but the results are visible in the panel E and panel F, indicating that there is an increase in the extracellular acidification rate whenever this VCAM-1 is overexpressed in the PANC1 and KPN2 cell lines. And about this lactate productions and the intracellular glucose levels, they are also upregulated with the overexpression of the VCAM. That can be seen here for the lactate production in the panel G. We can see that in the PANC1 and KPN2, the increased amount of lactate productions are statistically significant compared to the control case, and same results they have observed for the intracellular glucose level. That can be seen for the PANC1 and KPN2 cell lines. That is, their increased amount of intracellular glucose levels are higher compared to the control case. And apart from this seahorse XF analyzers and this lactate productions and intracellular glucose level, they also check the medium conditions, which gives us the clear indication of extracellular acidification rate. So when the VCAM is being knocked out using silencing RNA against VCAM-1, we can see that the color of the medium does not change, indicating that the pH does not change or does not become lower. However, when they overexpress this VCAM-1 using pcDNA 3.1, they can see that there is a color change. There is a color change in the medium, which could be usually because of the phenol indicator that are present in the medium, which turns yellow whenever the medium become acid, whenever the pH of the medium reduces, indicating that the extracellular acidification, indicating that there is an increase in the pH in the medium where this VCAM are being overexpressed for both PDAC-1 and KPN2 cells. And from the other seahorse analyzer results, it could be it indicates that this increase in the pH is because of increase in the cellular acidification rate, that is evident from panel E and F. Whereas in both the cases, in the control, we do not see a significant change in the color of the medium, indicating that this whole figure indicating that it's the Warburg effect that have been triggered in the pancreatic cancer cells with the overexpression of the VCAM that helps in the aerobic glycosylases, and that results in the aerobic glycosylases and other benefits that the pancreatic tumor cells observe, such as increase in the survival and the growth signaling pathways because of the Warburg effect are been are could be possibles in these pancreatic cancer cell lines with overexpression of the VCAM-1. So this is what they have presented in these results, or this is what they have made a point out of the observations that they get from these experiments. So I hope you understood how this VCAM helps in this Warburg effect of the PDAC cells. And these are checked on the in vitro, that is in the lab scales. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.